Hello everyone, welcome to MedMate. Today we are going to be talking about Physiology Module 1, Cell and Membrane, Transport through the Cell Membrane. In this lesson, you are going to learn about how transport of substances is happening through the membrane of the cells. And it's a very important lesson because you will use this concept later on when we will learn about action potential and overall how transport and hormones are working. So it's a very fundamental lesson. Without further ado, let's get started. So the first term that I want to explain for you is homeostasis. So homeostasis stands for the matter of fact that throughout our body and organs and our cells are trying to maintain a ionic composition. In order to be working optimally, they need to have these environments, these specific environments, and that environment is called homeostasis. Now, overall, we can divide our cells fluid into different sections, and the first section is intracellular fluid, which is inside our cells, and the other sections are interstitial fluid, which is out of our cells, and plasma, which is within our capillaries, and it's actually within our vessels. So, ionic composition of intracellular and extracellular fluid is also an important concept. You have to try to memorize which ions are more abundant out of the cell and which ions are more abundant inside the cell. That's because when you learn and when you memorize these, you'll then know what is the gradient for each of these substances. And then when we talk about transport, you'll learn that better. So basically, sodium is more abundant in extracellular fluid, as you can see here, and you can also follow up the numbers on the other table, on the other graph that I have illustrated for you here. So sodium basically is the concentration of sodium is 140 millimole per liter in extracellular fluid and 10 millimole per liter in intracellular fluid. The other substance is potassium. So potassium is more abundant in intracellular fluid, and the concentration of it in extracellular fluid is 4 millimole per liter and 140 millimole per liter in intracellular fluid. So what you have to mind is that sodium is more abundant in intercellular fluid. So the concentration of potassium is more abundant in intercellular fluid. So the gradient for potassium is from intercellular space to extracellular space. What about other substances? For example, here we have magnesium, which is more abundant in intercellular fluid, and chloride, which is more abundant in extracellular fluid. You don't necessarily need to memorize all of these numbers, but you have to know which molecules are more abundant in intracellular fluid and extracellular fluid. So transport is happening through our membrane, either via a channel, which can be which is that for example a protein channel, or it can happen through the plasma membrane itself if the molecules are fat-soluble molecules. The molecular structure of our membrane is it consists of a bilipid phospholipid layer. And it consists of a polar head and apolar hydrocarbon tail. So basically, we can divide types of transport based on the energy that they require to occur into two groups: passive and active. Now, passive transport is when we have movement of molecules and ion across a membrane from higher to lower concentration. So let's have a look here. Here you see passive transport, and here you see there are a lot of molecules in extracellular fluid and just a little bit of them in intracellular fluid. So anyways, the gradient is from extracellular fluid to intracellular fluid. The same thing happening here. There are a lot of substances here, there are a lot of maybe ions here, and their concentration is more in extracellular fluid. So the gradient is from extracellular to intracellular fluid. Now, this type of transport happens without energy, so it does not require metabolic energy, and it just happens down a concentration gradient. On the other hand, active transport is the movement of molecules and ions across a membrane from the region of lower to the region of higher concentration, and it always requires energy. So here you see, let's call these molecules cubic molecules, and these molecules, just there are two of them here in extracellular fluid, but there are a lot of them in intracellular fluid, and we want to take these two molecules inside the cell. For this action, since we are moving these molecules to the intracellular fluid and against their concentration gradient, we require energy. And while we use ATP, and in exchange of ATP, we can move this molecule to the intracellular fluid. Now, protein carriers, based on protein carriers, we can divide transport into carrier-mediated transport, which is facilitated diffusion and active transport. So we need a protein or a channel to conduct these kinds of transport, and non-carrier mediated transport, like a simple diffusion of ions through membrane channels, or simple diffusion of water molecules, for example, osmosis. One of the concepts that most of the students have problem with, or maybe they are a little bit confused with it, is the difference in between diffusion and osmosis. So here you see in these photos, there are a lot of molecules in this specific part of this glass of water. So diffusion is movement of molecules from higher concentration to low concentration. Both solute and solvent move. So both of them are happening, or both of them are moving at the same time. And so here you see there are a fewer concentration of the molecules. Maybe there's actually absence of these molecules in there. So these molecules tend to move to that part of the glass. While osmosis is when there is a movement of solvent across a semi-permeable membrane from high to low solvent concentration. So here we have our semi-permeable membrane, and you see there are a lot of molecules here, and there are just a few of them here. Now, this permeable membrane, this semi-permeable membrane, is only permeable to water. If it's only permeable to water, then water is the only molecule, water molecules are the only molecules that can move from this side to this side. Why? Because there are a lot of concentration of molecules here, so water tends to move from lower to higher concentration to make up for that difference in the concentration difference, and to get into equilibrium. Now, diffusion through a dialysis membrane, which is basically the same concept. So here you see we have this dish, and there is water in it, there is sort of a solvent in it, and we have this dialysis membrane, which is working like a semi-permeable membrane. But this dialysis membrane is only permeable to these red molecules. And these molecules, since their concentration is higher in this dialysis membrane, they tend to move from this dialysis membrane out to the solvent until they get into the equilibrium. We use this concept in real world and in treating our patients. So in dialysis, actually, we're using sometimes like we are using machines, which then makes up for hemodialysis. Maybe you have heard of that. Is when blood is channeled to an external filtering system. So here we get the blood from the patient. So it's a machine, and the blood is constantly circulating. The blood is coming from the patient, it's going to the device and to the machine, and then here we have a semi-permeable membrane. Now, you know, in those patients that are suffering from impaired kidneys, or their kidneys is unable to filter waste from their blood, we have to perform this procedure. And in this case, since we are actually from the other side of the machine, we are perfusing fresh dialysate into the machine, and this dialysate has a lower concentration overall. It has lower concentration of the substance that are available in blood. So those substances and waste tend to move out from the patient's blood to this dialysate, and this semi-permeable membrane is only permeable to those waste. So the clean blood is returning to patient's body afterward. Now, the other method, which is very interesting, and we use it also in clinical application, is that it's called peritoneal dialysis. And here, what we are doing is we have this dialysate, and we are introducing it into the abdominal cavity. And the person's own peritoneal membrane serves as the semi-permeable membrane. So what we basically do is that we are sending this dialysate into the peritoneal cavity, and here the peritoneal membrane is actually performing and is just exchanging the waste from patient's blood with the dialysate. And then we have a catheter installed, and this catheter drain all of those waste and all of those like dialysate, which was previously clean and now full of waste, back into the drain bag. So another concept is osmosis. We have actually spoken about it, but just like that there is a semi-permeable membrane which is only permeable to water, and then water tends to move from lower to higher concentration. Now, having this in mind, the pressure needed to just stop osmosis is the osmotic pressure of the solution. Why is it important? Because there is a clinical application to that. You know that patients that are suffering from cirrhosis, or they have impaired liver, or any sort of liver disease, and when the concentration of plasma proteins are low, because these plasma proteins are osmotically active, they are draining, or they are pulling out water from our interstitial tissue, so they don't let water to accumulate in our tissues. If the concentration of these plasma proteins are low, then a situation happens so-called edema. Now, edema is when it can be due to a lot of things, like, for example, inflammation or other things. But in case of those patients that are suffering from impaired liver or liver disease, what is happening, concentration of plasma protein is low. For example, albumin is low, and then water is moving from our vessels into the interstitium, and then it is staying and accumulating there. And as you can see here, there is an edema in these patients. All right, now another concept is molarity, osmolality, and tonicity. What is the difference in between these terms? Now, the only way that I think makes sense and can make it easier for you to understand is looking at how they are actually calculated. So basically, molarity, osmolarity, molality, and osmolality, all of them are measures of concentration. Now, molarity talks about the volume of the total solution, so solute plus solution combined together, and molality is about mass of solute and solvent separately. Now, let's have a look here. You see molarity, and in molarity, when we are calculating that, we put moles over 1 liter of solution. This is very important, solution. Meanwhile, when we are talking about molality, we are putting moles over 1 kilogram of solvent, not solution, solvent. Now, let's compare osmolarity and osmolality. Now, you see osmo, so we put osmoles here, and then we put 1 liter of solution underneath. This is osmolarity, very similar to molarity, and osmolality is when we put osmoles over 1 kilogram of solvent again. The same concept happened in molality when we had 1 kilogram of solvent. So the other term is tonicity. What is tonicity? The term tonicity is to describe effect of solution on the osmotic movement of water. So here, let me give you an example. Here we have red blood cells, and let's imagine we put this red blood cell into a hypotonic solution. What does hypotonic means? It means that there are lower concentration of molecules in these solutions. Now, what happens is that membrane is working like a semi-permeable membrane, like what happened in description of osmosis. Now here, water, if we place this red blood cell into this solution, water tends to move from this hypotonic solution into the red blood cell. Why? Because there is higher concentration of molecules inside the red blood cell. So what happens? Water continues to diffuse inside to get inside this red blood cell, and eventually this red blood cell bursts. And this process is called hemolysis. The other thing that we have to consider is placing red blood cell in a hypertonic solution. Hypertonic solution is when we have higher concentration of molecules outside red blood cells. So what happens in this case is that water from red blood cell tends to move out, getting into that solution. And what is happening to the red blood cell is that its volume is it's actually shrinking, it's getting smaller, and this process is called crenation. All right, now what is clinical application of this? So you know that we have different types of intravenous fluids, and they are generally isotonic to blood. What does isotonic means? Isotonic means that the concentration of molecules in this solution is similar to what we have inside our body, inside the cells of our bodies. Now, common intravenous fluids are normal saline and 5% dextrose. They are widely used in different types of like when we want to prescribe intravenous fluids. Other types of isotonic solutions, which is also frequently used in hospitals, is Ringer's lactate, and it contains lactate, sodium, chloride, potassium, calcium ions to more closely resembles the electrolytes of our body and in our plasma. So, but meanwhile, we have other kinds of intravenous fluid that are not isotonic. For example, mannitol, you can see the photo here. Mannitol is derived from the sugar mannose and is sometimes given intravenously because it is osmotically active, and it's prescribed to those patients that are having cerebral edema, and it helps with drawing water from swallowing tissue. In this case, as when there is a cerebral edema present in patients. For example, in patients that had trauma or stroke, we prescribe this intravenous fluid. Now, earlier on, we spoke about carrier-mediated transport. Carrier-mediated transport, in which net movement is down a concentration gradient and which therefore passive, is called facilitated diffusion. So, what is important here is facilitated diffusion is a passive kind of transport. And carrier-mediated transport that occurs against a concentration gradient and which therefore requires a metabolic energy is called active transport. A very important facilitated diffusion that is happening all the time is glucose, transport of glucose, and glucose is transported within our organs via these specific transport which are indicated and named GLUT. Now, there are different numbers of these GLUT channels. For example, GLUT1 is available in our blood-brain barrier, and heart. Is a high yield content because you will be asked these questions later on in different kind of examinations. So it's good to memorize them. GLUT2 is present in our liver, pancreas, small intestine, is also insulin independent. GLUT3 is available in our brain, neurons, and sperm, and is insulin independent. GLUT4 is available in our skeletal muscle, adipose tissue, heart, and also GLUT5, which is available in different tissues within our body. Alright, now, in epithelial cells of the kidney tubules and a small intestine, glucose is transported against this concentration gradient by a different kind of carrier, one that is dependent on simultaneous transport of sodium. So, in two parts of our body, the transport of glucose is happening via different types of transport, and we are using energy for transferring these kinds of molecules inside our cells. And is also dependent in transport of sodium. So now I told you about active transport earlier, so let's have a look. Primary active transport occurs when we hydrolyze ATP, and well, ATP is needed for these kinds of transport directly. Here you see we have potassium, and I told you potassium is more abundant inside our cells, but we want to move potassium from extracellular fluid to our intracellular fluid. What we are doing here, we are moving potassium against its concentration gradient. We need energy for that. Now, sodium is more abundant in extracellular fluid, but what we want to do is that we want to move sodium out of the cell. So what we are doing again here is we are pushing sodium against its concentration gradient. And for that, we require energy. Here we are using ATP to pull two potassium inside the cell and 3 sodium to push them out of the cell. This specific pump is called sodium-potassium pump. We call them pump because we are actually using energy for these types of transport. Another kind of pump is hydrogen-potassium pump, and here what we are doing is we are exchanging potassium with hydrogen. And again, because we are pulling potassium inside the cell, then we need ATP for this. We require energy for it. Another example is calcium pump, and what we are doing here is that we are pushing calcium out of the cell, and we are taking a proton inside the cell, and for this we also need ATP. So all of these kinds of pumps are active types of transport. Now, the steep sodium gradient is used to provide energy for the coupled transport of other molecules. So we need to maintain this gradient because we are having another type of active transport, which is called coupled transport. I'll explain about that in a bit. Now, the gradient for sodium and potassium is very important across our plasma membrane of nerves and muscle cells, because it's used to produce electrochemical impulses needed for function of nerves and muscle. So we also need to maintain that concentration gradient. And the active exertion of sodium is important for osmotic reasons, because if the pump stops, then the increased sodium concentration within our cells promote osmotic inflow. And you remember when we put this red blood cell inside a hypotonic solution, then water tend to move inside the cell, and that can continue until eventually the cell bursts. So, secondary active transport. Secondary active transport is, in this case, I'd like to start with an example. So this happens when we are actually moving one of these molecules, like it's a downhill movement of one of the molecules, and it's actually promoting an energy for an uphill movement of another molecule, which is moving against its concentration gradient. For example, here you see there are a lot of these molecules that are sodium, and you know that sodium is more abundant in extracellular fluid, and there are just a few of them here in intracellular fluid. What we are doing is that we are using this energy, this gradient for sodium. We are moving sodium, 2 molecules of sodium inside the cell. And what we are doing to glucose, you know that glucose is more abundant in intracellular fluid, but there are just a few glucose molecules here in extracellular fluid, and we want to get them, we want to absorb them. How we do it? We are moving glucose against this concentration gradient. Therefore, we are actually getting this energy for an uphill movement from movement of sodium, and we are getting glucose in via a movement of 2 sodium molecules. So these 2 sodium molecules are making that energy for an uphill movement for Golgi molecule. A great example of that is seen in epithelial cells in our intestinal lumen. Earlier, I told you absorption of Golgi in our intestinal lumen is via a sort of active transport. What is happening here is, you see here we have a intestinal lumen, and we want to move these Golgi inside our cell. What we do, we are getting two sodium molecules inside, and these two sodium molecules are making that energy we require for an uphill movement of Golgi, and Golgi is transported inside our epithelial cells. Meanwhile, this sodium is building up in this epithelial cell. For maintaining this concentration gradient, we need another pump to pump out these sodium and get the potassium inside. For that, we require ATP because we are moving sodium against this concentration gradient. So this type of transport indirectly using ATP. The pump itself is not using it, but for maintaining that gradient, we need another pump, for example here, our sodium potassium pump, to use ATP and push those sodium out of the cell. Now this Golgi is inside our cell, and via for example GLUT2 channels, we are absorbing Golgi into our blood. What is clinical application of that? So I'm sure you've heard about oral rehydration therapy. You know that in patients that are suffering from gastritis or sort of diarrhea or any sort of problem with their absorption, or when there is excessive loss of sodium and water, the patient is suffering from loss of water and dehydration. What we basically do, in some countries it's not practical to use intravenous therapy because it's expensive, but WHO years ago developed sort of new economical treatment which is called oral rehydration therapy, or ORT in another word, and quite by accident they understood, or they discovered this matter, that presence of Golgi aids the intestinal absorption of sodium and water. So we talked about it earlier, how Golgi and sodium are actually transported both together. So presence of Golgi is promoting absorption of sodium. Now let's answer some questions and review the topic. Simple diffusion and facilitated diffusion share which of the following characteristic? So simple diffusion does not require energy. Facilitated diffusion does not require energy. Let's have a look at the options. Number A, can be blocked by specific inhibitors, do not require adenosine triphosphate, require transport protein, saturation kinetics, transport solute against concentration gradient. So earlier on I told you neither of them require ATP, so option B is correct in this situation. Do not require adenosine triphosphate, ATP. Alright, next question. Which of the following best describes the changes in cell volume that will occur when red blood cell previously equilibrated in a 280 milliosmolar solution of sodium chloride are placed in a solution of 140 millimolar sodium chloride containing 20 millimolar urea, a relatively large but permanent molecule. Alright, so here the red blood cell is placed in, and is actually equilibrated at 280 milliosmolar solution of sodium chloride, and is placed in a solution of 140 millimolar sodium chloride. So sodium chloride, and we have two molecules here. We have to multiply 140 by 2, which gives us 280. Now, for now, this is an isotonic solution. But looking at the rest of the question, containing 20 millimolar urea adds up to that 280 millimolar solution, and it becomes 300 millimole. then this means that the solution is hypertonic. Now, what we have to pay attention to is that these molecules, the membrane of our red blood cell is permeable to urea. So it's written large but permeant molecule. So let's have a look at these options. I told you that the solution itself is hypertonic. When we put a cell in hypertonic solution, water tends to move out. When water moves out, then the cell starts to shrink. So we have two options here that show shrinkage. So option A and B. But the correct one is option B. Why? Because the cell's membrane is permeable to urea. So first it shrinks, but then since it's permeable, urea goes out to get to the equilibrium, so the cell returns to original volume. I hope that everything was clear for you. Hope you understand all part of this. If you had any question, please comment that below the comment section of this website, and our instructor get back to you and will explain anything that was not clear for you. I also suggest you to go to our Q bank and check your knowledge whether you've understood all parts correctly or not. So thank you so much. See you next time.